Hello everyone. Today's presentation in the Epigenetic and Cancer course is about novel epigenetic therapeutic strategies and targets in cancer. During this presentation, we will talk about the role of the drug development, يعني the repurposed drugs used in epigenetic inhibitors to treat cancer cells, how we can use the epigenetic editing in order to correct epi mutations at the level of the DNA or histones, the dietary compounds, how can they affect the epigenetic machinery of cells, the natural compounds also, how they can be used to treat cancer cells, the role of microbiota to induce or inducing epigenetic alterations in the host, targeting epigenetics by using terahertz, the cold atmospheric plasma as a treatment for the cancer cells epigenetically, and the role of nanotechnology to target the epigenetic machinery in cancer cells. Let's start by the first part, which is about the drug development as novel epigenetic inhibitor. First, developing a new drug from a scratch is a complex, long, and expensive method. On average, it takes more than 10 years, and with billions of dollars. And also the success rate, or the percentage of the success rate, remains very low at roughly 10 to 20%. So because of these challenges, researchers are looking for new approaches in order to speed up drug discovery and reduce the cost. And one such purposing strategy is the drug repurposing. So, what is drug repurposing? Drug repurposing, or DR, it's the reusing of existing drugs and new medical applications in different diseases. And drug repurposing are facilitated by different methods like network analysis, clustering, and text mining, where the data, our drug data, are classified in order maybe based on their mode of action, maybe based on the physicochemical properties of them, or any different criterion. Moreover, the repurposed drugs play a crucial role in the cancer treatment. Here you can see some repurposed drugs, and here you can see their original usage to treat the original diseases, and here the second you can see the second usage of the drug. يعني because of that they are used as repurposed drugs, يعني to treat يعني two different diseases. يعني they used an existing drug in a way to treat another disease. Now, epigenetic speaking, epigenetic pathways are common across different cancers. So, repurposed drugs can offer cheaper and more precise treatments for different types or certain types of cancer cells, like the case of repurposed drugs like apicedin, mahanin, berberin, etc. We're gonna discuss one of them only. Apicedin was originally developed as anti-protozoal agent to kill Plasmodium, which affects the hepatocytes and red blood cells. So when apicedin is taken, oh, we have a mistake. Has been. So apicedin has been originally developed as anti-protozoal agent to kill a plasmodium that infects hepatocytes and red blood cells. So when apicedin is taken, the epicidin enters the nucleus of the عفواً, to the nucleus of the plasmodium that are present at the level of the hepatocytes or red blood cells. When the epicidin enters على الcell بيفوت على it enters to the nucleus and to inhibit the activity of HDAC, keeping the histones acetylated, and the gene is يعني certain genes more expressed, and this will lead to the death of the plasmodium. From this point and later, researchers um, have discovered that apicedin can act as HDAC inhibitor in certain cancer treatment. So when it's taken, it will kill the cancer cells. So from here, we can say that apicedin is used as a repurposed drug. Now, the challenges that we can have for the يعني repurposed drugs that work at the epigenetic level. هون لا sorry, we can يعني here we can say that إنه the repurposed drug not only can act against HDAC, but also they can inhibit the NMT, or also they can inhibit BET, HMT, HAT, HDM, any epigenetic enzyme. And the challenge that we have is the specificity, because the epigenetic enzymes are widely expressed in the whole body cells. So we need to be specific by using our drugs. We need to يعني to target our cells, not any cell in the body, not any healthy cell. Now let's go to the second part, which is about epigenetic editing. What is epigenetic editing? Epigenetic editing is the usage of epigenetic enzymes in order to add or remove epigenetic marks either on the DNA or histones, in order to regulate the gene expression, without affecting the genetic code. For instance, when BRCA1 and BRCA2 tumor suppressor genes are genetically normal but epigenetically mutated. So in this case, the BRCA1 and 2 will not be expressed. Why? Because they are genetically normal but epigenetically mutated. So they are turned off, they are closed, مطفيين. Why? Because they are epigenetic mutated. And if we want to correct the mutations or the epi mutations, يعني المutations at the level of the epigene يعني epigenetically يعني الهستون او at the level of microRNA, or at the level of DNA, we can use epigenetic editing to correct the marks that can be put on the histones or the DNA. How can we do by this, by the epigenetic editing؟ هلا بنشوف. All we need is that Cas9 epigenetic editing، يعني epigenetic عفوا modulator coupled to the ADAT-Cas9, and we need a guard RNA that you can see here. This is the guard RNA, this is the DAT-Cas9, this is the epigenetic activator or repressor. يعني whatever it is, it can be any enzyme، يعني epigenetic enzyme. خليني اقول لك شو بدنا نعمل. First, the DAT-Cas9 binds to the DNA of interest, but when it binds, it does not induce the cleavage of the DNA. No cleavage of the DNA here. Why? Because it's that, because it's modified. لأنه the cleavage of the DNA creates an indel mutation, creates a mutation or frame shift, but نحن we don't want to create a mutation at the level of the genetic code. ما قلنا انه ال BRCA1 and 2 here are correctly genetically and they are correct genetically. We need to what? We need to correct the epi mutations. So because of that, we use the DATCAS9. The DATCAS9 is not able to cleave the DNA, just it bind here. هلا how it binds to this area not to this area, يعني not to this segment of the DNA أو here أو here. Why? Because the guide RNA هو complete complementarity بكون هو in a complementarity way to the sequence of DNA اللي بتكون 20 nucleotides. يعني شوف قديش في specificity يعني to bind to this area not to any other area. اوكي it's guided by the guide RNA. فاذا the guide RNA بيعمل guiding to target that Cas9 to sit on a specific location on the DNA. And now the epigenetic epigenetic active يعني effector which is the epigenetic enzyme can now do its job. Okay, so قلنا الجايد RNA هو to guide the that Cas9. that Cas9 بيعمل binding والcoupling للاكتيفيتر او الريبريسر هو اللي حيعمل epigenetic enzyme to correct the mutations. فهون the methyls are removed. Why? Because the epigenetic enzyme is removing these mutations يعني. And now the genes will restore their activity. كمان as I said ما في شيء هون نيو كله شرحته. Applications of epigenetic editing. Epigenetic editing can be used in researches and therapeutic potentials. In researches to understand the role of epigenetic changes in gene expression and disease development. In therapeutic potential to correct epimutations and genetic disorder. The challenges that we can face by using epigenetic editing هي الاميونوجينيسيتي لانه we can يعني introduce the DNA of the guard RNA of the epigenetic enzyme which is coupled to the DATCAS9 كلهم حيفوتوا in a DNA that can be put or engineered in a viral vector or a liposome. So these strategies will induce immunogenicity and inflammation also. We need to be specific in the delivery to overcome all the barriers in order to reach correctly كمان the cell that we want to correct it. Now let's go to the third part which is about epigenetic modulations by dietary compounds. Let's see the role of dietary compounds. How do they act epigenetically? Polyphenols. In this part we will it will be divided into two parts, polyphenols and fatty acids. Polyphenols, they are of plant origin compounds. They are natural compounds. They act as antioxidant and anti-inflammatory compounds. They are present in fruits, vegetables, tea, and herbs, and also in coffee. They modulate the action of HTAC, any enzyme. يعني they can act. They can modulate the activity of the epigenetic enzymes or the microRNAs. Here in this table you can see different types of polyphenols, and here you can see their origin, and here you can see their epigenetic effect. Let's take one example which is about hibiscus. Hibiscus is a plant used in salads and tea. It contains polyphenols acting on the يعني affecting the expression of microRNAs, different types of microRNAs, to reduce hyperlipidemia. Hyperlipidemia meaning the quantity of يعني lipids in blood. They reduce the high quantity of cholesterol and lipids in blood يعني, and preventing the يعني they reduce the risk of also of atherosclerosis. هلا hibiscus has four مثلا many polyphenols. four of them عندنا هولي anthocyanins بيعمل reduces the microRNA المير 33 at the level of the hepatocytes and macrophages to increase the cholesterol efflux يعني to increase the entry of the cholesterol from the blood into the hepatocytes to not keep the blood rich in cholesterol, and also decrease foam cell formation. Foam cell formation in the macrophages when they phagocyte الكوليسترول in blood بيعملوا هيك like a foam to form plaque، يعني هو بصيروا atherosclerosis plaque، فهو بخففهم. flavonoids هون اكيد هيدي this one الفوم cell we are talking about يعني in macrophages. كمان reduces mir 121 to reduce lipogenesis and triglyceride synthesis at the level of the hepatocytes and many etc. Here كمان also about hibiscus يعني حنشوف كيف ال mode of action of anthocyanin عم يعمل reduction 133 promoting ABCA1 ABCG1 to reduce هيدا by reducing قلنا okay inducing ال HDL formation in hepatocytes and cholesterol etc. Now let's go to the second part, which is about the fatty acids and the epigenetics. How can the fatty acids affect epigenetics? Now let's talk biochemistry, structural biochemistry about the four types that we have about fatty acids. We have polyunsaturated fatty acids, which means they have many double covalent bonds in their chain. They are found in fish, nuts, and seeds. They regulate DNA methylation and histone acetylation. Monounsaturated fatty acids, they have only one double covalent bond, like the fatty acids that are present in olive oil and avocados. They influence the expression through histone modification. Saturated fatty acids meaning عندهم no double covalent bonds found in animal fats and dairy products associated with the pro-inflammatory epigenetic changes. Finally, trans fatty acids, they are industrially produced fats. They alter the DNA methylation in a way that promotes metabolic disorder. Now let's talk about the omega-3 fatty acids and epigenetic effects, their beneficial epigenetic effects. Omega-3 means they have a double covalent bond between the third carbon and the fourth one at the methyl extremity, يعني not at the acyl extremity, at the level of the methyl extremity. فإذا this is the first, second, third. هيدا between third والfourth. هيدا omega 3. It's called alpha linoleic acid. Alpha linoleic acid having 18 carbon atoms with 3 unsaturation. This is the first, second, and third. عشان هيك اسمه omega 3. And this one is an omega 3 اللي هو eicosapentaenoic acid. كمان having this one has 20 carbon atoms with 5 unsaturation. Why it's called omega 3? لأنه the first double covalent bond at the extremity of the methyl group هي between the third and the fourth. وهيدا docosapentaenoic acid كمان it's omega 3 لأنه 3. These fatty acids, omega 3 fatty acids, influence DNA methylation and histone modifications in gene involved in lipid and glucose metabolism and inflammation. Here we can see the role of the omega 3 fatty acids epigenetically. They induce the DNA methylation, like reducing the methylation of fat oxidation genes like PPAR alpha and CPT1A. So they reduce the methylation, which means they activate or increase the activity لأنه less methylated يعني more active, increasing energy expenditure. Now histone modification, increasing H3 acetylation at PGC1 alpha. So when it's more acetylated معناتها, which means more active, enhancing mitochondrial activity and reducing fat storage. And at the level of the MIR33 هون, these epigenetic modulations act as anti-obesity, and these also epigenetic modulations lower the risk of diabetes mellitus type 2. Okay, هون أكيد we are talking about omega 3 fatty acids. كمان these epigenetics lower the risk of atherosclerosis. فإذا they lower the risk of atherosclerosis, they lower the risk of diabetes mellitus type 2, and they act as anti-obesity. يعني شو بدنا أحلى من هيك؟ 3 أشياء. الأوميجا 3 كتير مهم و it's present in fish. لازم نركز على الفيش بهيك حالة. Now let's go to what نحن وين صرنا. Okay. Now let's see the Eskimo studies. Omega 3 consumption by يعني the results of يعني of research done on Eskimos. The Eskimos who consume a high amount of fish, so they consume high amount of omega 3 compared to the American residents, and the results show increase in or decrease, sorry, in the obesity, chronic diseases such as diabetes and heart disease. مثل ما قلنا انه الاوميجا 3 حيخفف، ومثل يعني مثل ما شرحنا قبل عمل ديكريز كمان لقوا انه هايبوميثيليشن للفات اسيد سنتيز، يعني حيكون عنده اقل ابوبتوس انفولد، اقل ليبيد ميتابوليزم ريجوليشن، وهايبرميثيليشن سايتس لل AHRR. هايبرميثيليشن معناتها هيدي اتس سايلنسد، معناتها for better glucose tolerance and insulin sensitivity. Here in this research you can which is done or conducted on females who consume more olive oil and avocados اللي هن rich in omega 6 not omega 3. يعني double covalent bond from the method بتكون between the 6 وال 7s. Hypomethylation of CPG sites of gene TNF alpha. Hypomethylation less methylation معناتها it's more active، يعني it will induce inflammation linked to obesity and metabolic issues. يعني this one is not يعني منه كتير healthy to consume a lot of omega 6 يعني. Here you can see the results, the epigenetic results done by saturated and trans fatty acids. Now let's go to the fourth part, which is about the natural cancer treatments that target epigenetic processes. So several natural compounds present in fruits, vegetables, and herbs have been discovered. Um, um, have been discovered to act as natural compounds and as epigenetic modulators in different cells by modifying the DNA methylation or affecting histone modifications or regulating non-coding RNAs. Here you can see كمان مثل قبل polyphenols والsource their source، وكمان هون their epigenetic target والeffect on the cancer، يعني on cancer progression أكيد بخففوا. Natural compounds combat cancer through they fight cancer through يعني tumor suppressor gene reactivation, so demethylation of gene like P16 and P53, oncogene suppression, which means silencing the genes like c-Myc and RAS, cell cycle regulation, induction of G1S or G2S arrest, etc. Let's see the effect of curcumin, which affects the expression of different types of microRNAs to stop the AKT, for example, or to stop BCL2. So in this case, they affect the c-Myc. Okay, they reduce the activity of c-Myc, so the curcumin reduces, which is a natural compound, reduces the progression or cancer progression or aggressiveness. Here in this figure or illustration, we can see the effect of curcumin also. But before talking about the role of curcumin, let me explain what we have here. The P10 is a tumor suppressor protein which stops the activity of PI3 kinase to phosphorate AKT and and stopping the يعني الaggressiveness أو progression of cancer. فإذا P10 هو essential to stop the activity of PI3K to stop the show phosphorylation of AKT1 لأنه when it's phosphorylated it will induce the progression of cancer. So P10 stops this activation, and in this case it's very essential. It acts as tumor suppressor protein. How can P10 is affect or is inhibited? These microRNAs can bind to the messenger RNA of the P10 at the 3' UTR with incomplete complementarity. يعني any one of them مع incomplete complementarity على السيد region بعلق على المسنجر RNA of the P10, and this will lead to the degradation of the messenger RNA or translational repression. So in this case لما يعلقوا any one of them على المسنجر RNA, we will not have enough quantity or no P10 protein now. ما في protein أبدا. So the PI3K is always active, is always phosphorate AKT, وعنا always progressive, وعنا aggressiveness عنا بيكون في aggression للكانسر. So microRNAs here, these microRNAs are what are يعني we need to stop their activity. We need to eliminate them. How can we eliminate them? بالكركمين. فإذا curcumin بيعمل inhibition transcription of these microRNAs. In this case, the P10 is in the normal state. It's not degraded. It's present in the cell, and it can stop now the activity. لما بياخد signal, well, it takes a signal to stop the activity of PI3K. It will stop it and prevents the يعني phosphorylation of AKT. Now let's go to the fifth part, which is about the induced epigenetic alterations in the host. Let's start by the gut microbiota and their role as an epigenetic regulator. خلينا نقول بالأول إنه first the human body is home of trillions of microorganisms known as microbiota that live in the gut. Okay, and these microbiota are responsible for the fermentation of many substances present in the food to produce many metabolites like the short chain fatty acids, polyphenol metabolites, vitamins, tryptophan metabolites, and polyamines, acting as epigenetic modulators يعني to affect the DNA, histone, or non-coding RNAs regulation. Here we can see the formation of propionate. What is propionate? It's a short chain fatty acid produced from the fermentation of the microbiota of certain substances like hexose, pentose, fructose, and raminose. So this is the metabolic pathways that can the microbiota take, maybe by this one or this pathway or this pathway based on the يعني species, the type of the species of microbiota based the enzymes that they have. Here we can see the fermentation of amino acids will lead to the formation of propionate or butyrate, depending on the type of the species and the enzymes that they have, metabolic enzyme that they have. Now let's see the function or the role of butyrate in epigenetics. The butyrate hypomethylate line 1 and FFAR3 genes أكيد هو indirectly بيعمل hypomethylation. Hypomethylation means removing of methyl groups على promoters للcytosine للpromoters. This will lead to the reactivation of these genes, leading to the reduce of obesity, insulin resistance, and other metabolic disorders. وهذا شيء مفيد يعني. Butyrate acts as HDAC inhibitor to increase the H3K27ME3 enzyme. هيدا حيعمل methylation هيدا الانزيم. فإذا HDAC is inhibited يعني هيدا it's more expressed. هيدا الانزيم is more expressed. هيدا it's an epigenetic enzyme حيعمل methylation لأن NF-kappa B gene promoter. حيقفلها لهيدي الجين انف كابا بي اللي هي مسؤولة عن الانفلاميشن بالانتستين، وهيدا شيء كمان كتير منيح etc. Here we can see the butyrate and FOXP3 relationship between them. The butyrate is a short chain fatty acid. FOXP3 هو a gene for للinflammation. It's anti-inflammatory property عنده. فإذا wild type mice were treated orally مع 100 millimole of sodium butyrate for 3 weeks. عم نشوف انه significant increase of in يعني in FOXP3 plus Tregs in the tissues after butyrate treatment. شفنا only مش بالother immune tissues، بس شفناها بالكولن، لانه ليكو كيف عملت increase للفوكس بي 3، معناتها انه البيتريت عم يعمل يعني more expression to this gene. It affects يعني untreated كان 12.6 مع البيتريت طلع 1.7، معناتها it amplifies this gene how by HDAC inhibition. فإذا هيدا الجين صارت more acetylated. Let's see if butyrate can affect the IFN gamma gene بال TH1 و 17. عطيني هون شو ما في increase أبدا، معناتها ما بيعمل expression لها، ما بيعمل affecting epigenetic loss، مو بفيدها. Now let's go to the following part, which is about the targeting epigenetic using terahertz radiation. Terahertz radiation he reveals to electromagnetic waves with frequencies between 0.1 terahertz to 10 terahertz corresponding to a wavelength range of 1 millimeter to 0.1 millimeter. It's a non-ionizing radiation, which means it lacks the sufficient energy to eliminate electrons from atoms, and it's non-thermal radiation, which means it does not increase the temperature where it's reacting. Terahertz wave can penetrate deep into tissues without damaging or without damages. It induces epigenetic modifications like DNA methylation. تعمل methylation للجينز، يعني it increases the methylation of genes. يعني هون in this case شو it reduces the expression of some genes at the level of many cells. Also it يعني increases the quantity of reactive oxygen species بالcells، وهيدا reactive oxygen species will damage the cell based on the intensity of the terahertz، حسب قديش الانتنسيتي. فاذا شو قلنا بتعمل methylation للدي ان اي، يعني بتعمل lowering للاكسبريشن لسام genes، بتعمل increase بالquantity of reactive oxygen species يعني. Here you can see the NRF2 which is the master regulator of antioxidant response. شو هو ال NFR2؟ NFR2 مش FFR2، NRF2 stands for nuclear factor erythroid 2 related factor. انه it's the master regulator of antioxidant response. As you can see here عنا oxidative stress. It will induce the NRF2 to be activated here يعني بتفكوا عن هيدا بأنه بيعملوا inhibition. And this when it's phosphorylated it will go to the nucleus to activate some genes. It acts like يعني هو مثل promoter أو it's like a transcriptional factor to induce the release of enzymes called antioxidant enzymes like the HO1. And these enzymes, antioxidant enzymes, will reduce the oxidative stress the cell will stay alive. يعني it's very essential يعني it's not present ما في these enzymes will not be present يعني. وهون as we said انه بالnormal cells ال NRF2 حيعمل release للانزيمز اللي هن antioxidant enzymes. These ones or these antioxidant enzymes will reduce the يعني reactive oxygen species, and this will lead ultimately to the oxidative stress protection. But when we use TR يعني terahertz, terahertz هتعمل ميثيليشن لل للبروموتر تاع ال NRF2. يعني ال NRF2 will not be، يعني صارت الجين تاعيتها هي البروموتر at the level of the promoter is methylated، يعني it will not be expressed، ما في عنا NRF2. معناتها in this case antioxidant enzymes will not be produced أو synthesized يعني. فإن this case الرياكتيف oxygen species حيصيروا increase بالسل، والكيموثيرابي حيعملوا مع التيرا هرتز حيعملوا increase بالار او اس، وهيك this will lead to the show oxidative mediated cell death. Cancer cell will die يعني. Here we can see the effect of the terahertz on cells. So when we increase the power of the terahertz we can see إنه هون late apoptosis will increase، early apoptosis will increase، وكمان necrotic cells كمان will increase. يعني this will lead this will show إنه التيراهرتز can kill can يعني lead to apoptosis أو kill the cells، و some of them حيضلوا survived حيعملوا cell survival modulation، و some of them حيعملوا growth أو rest ما حيكملوا their growth. هلا اكيد السينرجستيك effects of terahertz wave with ultraviolet radiation او epigenetic drug او chemotherapy agent will increase the effectiveness of the therapy. يعني هتكون اكتر يعني لما اعمل combination by two مثلا two ways، يا بشتغل يو في مع تيرا هرتز او epigenetic drug مع تيرا هرتز او كيموثيرابي مع تيرا هرتز، كلهم حيعملوا حيقوى اكتر هيدا potential for enhanced cancer treatment outcome. Now in this part we're gonna study about the cold atmospheric plasma. Plasma when ionized generates reactive oxygen species and reactive nitrogen species. So plasma when ionized generates reactive oxygen species and reactive nitrogen species, and this method will be generated under cold conditions. This method is used in cancer therapy. هلا the cap induces oxidative stress in cancer cells by ROS and RNS. So reactive oxygen species and reactive nitrogen species will induce the cell in the oxidative stress. Why? Because these species اللي هن ROS أو RNS مثل hydrogen peroxide, nitric oxide أو superoxides will cause the DNA damage or protein oxidation or lipid peroxidation, and this will trigger the cell to go toward toward apoptosis. And why the cap is will kill more the cancer cells compared للhealthy ones، لأنه الكانسر cells عندهم more oxidative stress و less level of antioxidants مثل الانزيم اللي شفناهم قبل. So reactive oxygen species اللي عم يجوا generated from هيدي شو اسمها الكاب. Okay, الcap treatment will increase the ROS, وال ROS will take the cell to go up toward apoptosis. كمان الcap will induce قلنا ال ROS وال RNS that will كمان interact مع ال epigenetic modifiers مع الانزيمز تاعون الهيدا reactivating tumor suppressor genes أو inactivating proto-oncogenes. And this can also modify the expression of tumor suppressor microRNAs or microRNAs. يعني ال ROS كان مثل ما قلنا مش بس بتشتغل مش بس بتشتغل at the level of the DNA، بس كمان بتشتغل at epigenetically، بتعمل mutations، بتعمل ROS RNS، بيعمل DNA damage، بيعمل epigenetic modifier بغير لهم activity، بعلق عليهم فبتشتغل in two ways. هون we can see that شو انه in the case of breast cancer cells بهيدا التايب ال MCF7 breast cancer المير 19A بيلعب دور to stop the translation لل P10 to يعني بيعمل degradation للمايكرو RNA لل P10. And in this case, the P10 cannot stop the activity of PI3 kinase to phosphorylate AKT. In this case, the AKT is always active, and this will lead to the proliferation, metastasis, and chemo resistance. But when we do cap will يعني بيعمل methylation للبروموتر اللي بيعمل expression للمير 19A3P. So in this case it's not expressed وهيدا ال P10 is present، ما حيكون degraded messenger RNA تبعه، وحيعمل inhibition وما في عنا proliferation، ما في metastasis، ما في chemo resistance. Here you can see the potential benefits of CAP. هي non-invasive, selective cancer cell killing, induces apoptosis, etc. Now let's go to the last part, which is about the role of nanotechnology in fighting and targeting the cancer cells epigenetically. Nanotechnology هي enhances drug stability and ensures drug delivery. يعني it protects drugs from degradation, maybe from enzymes or interaction مع proteins أو to go to يعني not specific place. It increases the bioavailability يعني it stays for a long time in the body, it will not be eliminated rapidly. reduces toxicity يعني ال side effects to not يعني bind to non-specific cells. Here you can see the nanoparticles. Nanoparticles they can enter to يعني they can enter to the host cell that we want to target it. They can work في منهم هن بيكونوا specialized to induce the formation of ROS و RNS. the cell will go up through apoptosis، أو ممكن يشتغلوا at the level of the expression of يعني بيشتغلوا على المايكرو RNA، ممكن يعملوا degradation للمايكرو RNA، ممكن يعملوا dysfunction بالميتوكوندريا and this will lead كمان ليروحوا على الابوبتوسيس by releasing more ROS أو RNS. كمان ممكن يشتغلوا على الجينز يعني على epigenetic enzymes، ممكن يعملوا activation أو deactivation، وهون الاكسبريشن للجينز حتكون تغيرت أو لأنه الاسيتيليشن حتتغير أو الفوسفوريليشن للهيستون أو الميثيليشن للسيتوزينز أو أي شيء. هون you can see the epigenetic effects of nanomaterial مثل السيلفر لما بيكون AGNP أو Gold AUNP كيف حيعمل micro RNA dysregulation to alter gene expression. مثل nanoparticles حيعمل هون leading to abnormal gene regulation، هون disrupted cell cycle, inflammation, cancer progression. وهيدا beneficial for cancer therapy but may cause toxicity in healthy cells. يعني هيدا عنده disadvantages. Cell cycle disruption, DNA mutations, potential cancer risk by using this one. Here the second generation حتكون by using epigenetic drug, like instead of using nucleoside analog اللي هن more stable better tolerance مثلا ديسيتابين. ديسيتابين هو it's an DNMT inhibitor و it's not stable, so we can use guadicitabine which is more stable than this one. So when we use the second generation بكون عم نعمل بالstability we are increasing the stability to not be degraded easily by enzymes. هونيك ما حيعمل degradation. one nanoparticles بيكونوا عم يعملوا covering للepigenetic drug والchemotherapy together. يعني هون we are working synergistic therapy, we are doing synergistic therapy. يعني عم نعمل covering للepigenetic drug والchemotherapy together by nanoparticle. مثل الكيس of lipid nanoparticles can encapsulate both ال DNMT inhibitor and chemotherapy. doxorubicin هون DNMT inhibitor فينا we can use second generation اللي هو شفنا هون اسمه guadicitabine. So this will lead to this will improve the drug uptake by tumor cells and enhance anti-cancer effect. وfinally the future directions حتكون the future researchers are aiming to optimize يعني nanoparticle تعمل encapsulation ل siRNA small interference RNA for targeting cancer cell death. لأنه siRNA أقوى بيعمل complete complementarity وبيعمل knock down للجين. ف in this case the cell will go دغري directly to apoptosis. بس هي specificity هي the challenge that we have. Thank you. يعطيكم العافية.